Welcome back to The Explainer. Today we're jumping straight into section 6.3. How is sound intensity represented in the auditory nervous system? Now, on the surface, you might think, well, that's obvious, right? One of the most basic properties of our auditory nerve fibers is that as a sound gets louder, they just fire faster. More sound equals more electrical spikes per second. Simple enough. But as we're going to see, the actual biology reveals a massive, almost mind-bending paradox in how our nervous system actually pulls this off. It is absolutely not that simple. To solve this biological puzzle, we've got a clear roadmap for today's explainer. First, measuring intensity discrimination. Then we'll hit the dynamic range problem, explore coding by spread of excitation, look into coding by phase locking, and wrap up with the dynamic range solution. All right, let's dive into part 1, measuring intensity discrimination. So before we can unpack the internal neurology, we need to establish exactly how scientists measure our outward ability to tell two sound levels apart. And the baseline measurement for this is called the Weber fraction. In auditory science, this is used to calculate the just noticeable difference. Essentially, it's the precise tipping point where a listener can reliably detect that a baseline sound has gotten just a tiny bit louder. It's usually expressed as a ratio or in decibels. So to put that into perspective, for a wideband noise, the Weber fraction is roughly negative 10 decibels. That basically means you can just barely notice a 10% difference between the intensities of two wideband noises. And the way scientists actually measure this in the lab is pretty clever. They present a listener with two sounds separated by a really short delay. The first interval plays the baseline sound. Researchers call this the pedestal. Then the second interval plays that exact same pedestal sound, but with a slight increment in intensity tacked on. The listener's job? Just pick the louder sound. Over a bunch of trials, researchers keep shrinking that increment down until the listener can just barely tell the two sounds apart. Which brings us to part 2. The dynamic range problem. Okay, so now that we know how to measure hearing performance, we run headfirst into a giant physiological paradox. When scientists actually tested listeners with different kinds of sounds, things got weird. With wideband white noise, the Weber fraction stays totally constant across a massive range of volumes. That's a textbook example of Weber's law. But pure tones, they behave completely differently. They show what's known as a near miss to Weber's law. Basically, instead of staying constant, our ability to tell pure tone intensities apart actually gets better as the sound gets louder. When you look at the actual data for pure tones, you see this distinct downward slope. That's the near miss in action. It shows our discrimination performance improving as the pedestal level cranks up, going all the way up to 100 decibels. And listeners can easily keep telling intensity differences apart, even up to 120 decibels. And right here is where we hit the wall. Here is the big dynamic range problem. If human listeners can brilliantly discriminate pure tones up to 120 decibels, how is that physically possible when the vast majority of our auditory nerve fibers completely saturate and max out their firing rates at just 60 decibels? Literally, most of your neurons physically cannot fire any faster once a sound hits that 60 decibel mark. They're totally tapped out. So how on earth is your brain detecting subtle volume changes at 100 or 120 decibels? Let's look at the first theory in part 3, coding by spread of excitation. To explain how we process those booming 120 decibel sounds when our main neurons give up at 60, scientists came up with this idea. The theory goes, at low levels, only a tiny patch of your basilar membrane gets stimulated. But as a sound gets really loud, that excitation kind of spills over into adjacent neurons. As the level hits 80 or 100 decibels, the neurons tuned exactly to that frequency are totally saturated. They've hit their limit. But the adjacent, higher frequency neurons, the so-called skirts of the excitation pattern, they are getting less direct stimulation, so they haven't saturated yet. The thought was that our brains are smartly pulling intensity data from these unsaturated neighboring neurons to figure out just how loud a sound is. It sounds like a rock-solid theory, right? Well, scientists had to test it. They set up an experiment to deliberately block the brain from using those adjacent neurons. To do this, they played the pure tone alongside a band stop noise. Think of this as a background hiss that has a notch carved out of the middle right where the pure tone sits. This background noise essentially masked those excitation skirts, completely jamming the signal from the adjacent neurons. Now, the logical expectation here was that without those extra adjacent neurons, a listener's ability to tell loud sounds apart would just fall off a cliff. But surprisingly, no. Even with those excitation skirts completely masked by noise, human listeners were still incredibly good at discriminating intensity at high levels. The near-miss effect did disappear, sure, but the overall performance stayed highly accurate. So this proved that while the spread of excitation might help a little bit, it is definitely not the whole story. Our brains are clearly doing something else. Moving on to part 4, coding by phase locking. Since the first hypothesis didn't fully pan out, let's look at another angle. This second theory argues that our nerves don't just communicate by how fast they fire, but by when they fire. The idea is that even if a nerve fiber's overall firing rate is totally maxed out, it can still represent louder sounds by synchronizing its firing pattern more and more tightly to the dominant frequency of the sound wave. The brain then theoretically reads this increasing regularity, this phase locking, as an increase in intensity. It's an elegant idea for sure, but unfortunately, it has a fatal flaw. Phase locking is a very real, well-documented phenomenon, but we know for a physiological fact that it completely breaks down at high frequencies. The neurons literally just cannot fire fast enough to lock onto the super tight waveforms of high-pitched sounds. And yet, human intensity discrimination works flawlessly at high frequencies. So if phase locking stops working at high pitches, but our volume detection doesn't, then phase locking just cannot be the primary way our dynamic range works. Which brings us to part 5, the dynamic range solution. Okay, let's recap the mess we're in. Our primary nerve fibers max out at 60 decibels. The spread of excitation theory? Insufficient. The phase locking theory? Fatally flawed at high frequencies. We need a way out of this paradox. Enter researcher Neil V. Meister. He ran a super detailed statistical analysis of auditory nerve activity. And brilliantly, the solution to this seemingly impossible biological bottleneck comes down to combining the data from just 50 specific neurons. See, V. Meister focused on a minority class of neurons known as low spontaneous rate fibers. Now, these fibers have really wide dynamic ranges, but on their own, their individual responses are shallow and, frankly, pretty noisy. By themselves, they don't seem very useful. But V. Meister mathematically proved that if the central auditory system just pulls the information from about 50 of these specific fibers tuned to the same frequency, all that random statistical noise basically cancels itself out. When you mathematically map out a pool of just 50 of these neurons, it generates a predicted Weber fraction curve that perfectly matches actual human performance data up to extremely high decibel levels. So, the whole mystery is solved through ultimate biological teamwork. Most of our fibers max out early, right? But then, this tiny minority of low spontaneous rate fibers steps in to take over the heavy lifting at those high volume levels. By pooling their noisy signals together, the brain dramatically reduces the statistical variability, giving us a crystal clear picture of the volume. Plus, all of this beautifully exploits the physical compression happening right on the basilar membrane itself, which squishes those massive physical intensities down into a totally manageable range of nerve firings. So we now know for sure that our auditory nerve genuinely packs enough firing rate information to perfectly account for human hearing across our entire dynamic range. But V. Meister's math leaves us with one final, incredibly provocative plot twist. His analysis showed there's actually way more firing rate information available for low-level, quiet sounds than for loud ones. So the real question we should be asking isn't why we're so good at hearing loud sounds. It's actually, if our peripheral auditory nerve is sending so much perfect, highly detailed information up to the brain, why is our central nervous system so surprisingly bad at using it to interpret very quiet sounds? It seems the real limitation isn't in our ears at all, but hidden somewhere much deeper in the brain. And you know what? That is a fascinating mystery that we'll just have to save for another explainer.